Hello and welcome to this video that goes through the Exam Pro plant hormones questions. So question 1, hormones called auxins control plant growth. A student investigated plant growth responses in roots. He uses a method which involves growing 3 bean seeds until their roots are 1 cm long, attaches the 3 bean seeds to moist cotton wool in a petri dish, and then each bean seed root should point in a different direction. Fixes the petri dish vertically for 2 days in the dark and shows the results. So the first thing it wants to do is describe the direction of the growth of the bean roots after 2 days. So the direction of the roots, you can see that this is the root, and we can see that it's extended downwards for this one. We can see that this one has bent round, and this one has been actually quite the biggest change, so it's come all the way down. Now the reason why we have this change, the direction, so first of all our first one is what's the direction of the root growth? And the direction of the root growth is that it's growing down, and you can see that without actually knowing any biology. You can see that visually. Now the reason why is because it's going towards gravity. Now why? And this is actually the correct biological term is gravitropism. So anything to do with the roots is to do with gravitropism. But why? Why does this happen? Well, at the end, at the tips of the plants, at the tips of the roots, and also the tips of the stems as well, but at the tips of the roots, auxin grows. And this is what has essentially is a hormone that allows the plant. It's a growth hormone for the plant. But when the plant is when it's in the roots, what happens is that gravity is acting down. So our auxin grows all the way down, and gravity pulls essentially the auxin downwards to the bottom of the root. Now, where the auxin gathers, that's where the plant grows. So what happens is that the auxin grows more in that one. So we get auxin that's like here, and that causes the plant to bend around if it's being horizontal and grow downwards. So you can think of it as the plant always grows in the direction of the auxin. In the roots, this is because the gravity pulls it downwards. So gravity forces all the auxin to the bottom of the root, and then when it starts to grow, this is the part that elongates and gets bigger. So that explains why roots will always grow downwards, because the auxin is being pulled down by the gravity, and therefore the plant will grow with the auxin where the auxin is located, and therefore grows downwards, and eventually will be a straight line. So eventually the root will look like this. So whenever it talks about roots, it always goes downward due to gravity. Question 2, the student then noticed shoots growing from the seeds. He then put a light above the petri dish but did not move the seeds, and allowed the seeds to grow for 2 more days. Predict the direction of the bean shoots after 2 days. So the direction of the bean shoots, they should grow upwards. And the reason why is they're heading towards the light. Why does this happen? Well, let's say we have the sun shining, and we have a stem here that has auxin. What happens is that you can think of it as like auxin is allergic to light. It doesn't like light at all. Essentially, the sun interacts with the auxin. The mechanism is not 100% confirmed, and you don't need to know the mechanism behind or any of the mechanisms to do with it. But you can think of it as the sun doesn't want, the auxin doesn't grow on the side of the sun, it grows on the shaded side. So what happens is that as the plant grows, almost like the plant will grow this part first, and then follows up at the bottom part. So what happens is that the weight of that part bends the plant over, so it bends over to the right. So therefore the result of the plant is that it grows towards the sunlight. So again, the auxin is going to grow away from the sunlight, so in the shady part, so then that causes the plant just to grow upwards, and it bends towards it simply because the uxin is going to cause the plant to become heavier on one side. That bends it towards the light. And again, you could have it, if the uxin is on this side, it grows upwards here first, which bends, and if the light is here, so again it's always pushing it towards the light when it grows. Remember, uxin grows where the plants grow where uxin is, because it's the growth hormone, and uxin doesn't like the light, so essentially it gets forced onto one side. Okay, let's moving on to part C. Ethene is a plant hormone, and ethene causes fruit to ripen. Scientists measure the concentration of ethene found in fruit at different stages of ripeness. At what stage of ripeness is there the most ethene? At what stage of ripeness is there most ethene? So essentially it's at stage 3. The highest value here, very straightforward question. Concentration is the highest, so this is stage 3. Suggests how the scientists can find out if the result for stage 1 was an anomaly. So if you want to, anytime you see an anomaly, this is a result that is not in keeping with the rest of the data. You think that there's an error. So what we have to do is we have to repeat the experiment. So any experiment repeats will improve your chances of determining if an anomaly is actually an anomaly or is just a mistake that you've made or a mistake in the equipment. So repeat the experiment. Apologies for the pronunciation. Gibberellins are a different type of plant hormone. Farmers growing cotton plants in cold climates sometimes soak their seeds in a solution of gibberellins before planting the seeds. So Giblin Solution, gibberellin is another hormone, and this stimulates plants to grow. So it's another growth hormone, and what it does is that the seeds will germinate sooner. So usually seeds have to go through a process where they need the correct conditions. Once the correct conditions are met, then the seeds will begin to germinate. But essentially, if we use gibberellin acts as like a growth hormone, so therefore it will ensure that all the seeds in the batch germinate at the same time, and they can germinate sooner as well. So why is this good? So seeds germinate sooner because it's a cold climate. You're not going to have as long growing season. Your summers are going to be quite short, so the seeds germinate sooner to allow a longer growth, or allow a longer period to grow. Okay, and that's question 1. Question 2, a student investigated growth in plants. The student planted a seed in damp soil in a plant pot, put the plant pot in a dark cupboard, and the image shows the results after 5 days. So again, after the 5 days, the roots had grown, so the roots always grow in the direction of the force of the gravity. Again, that's because uxin is produced. So therefore that's what causes it to grow in that direction. After the 5 days, the shoots had grown, and again gravity is acting downwards and this is going upwards, so essentially it's against the force of gravity. Some mistakes, common misconceptions. So away from light, well, that's not correct because there's no light here, it's inside the soil. And then secondly, towards water, well, the soil is all damp, so you're not growing towards any water, because we can assume here that there's equal amount of moisture inside the soil across. So don't fall into the trap of thinking about water. Roots and shoots are dependent mainly upon light and uxin and gravity. So roots are gravity controlled, shoots are mainly light controlled. After the plants are grown, the student put the plant pot by a window with lots of light. The illustration below shows this. So complete the diagram to show the appearance of the student's plant after 20 days by the window. So what we should have is, again, in this situation here, the light is coming in on one side, so the uxin will be produced on this side, so therefore it's going to go and bend towards the light, like that. So when we've got the appearance plant after a few days, you're going to have it bent towards the light, like this. And we can do a thing. Again, the drawing does not have to be perfect, but it does need to be shown drawing going leaning towards the window. Explain the advantage of the plant of growing in the way that you have drawn in part BI. So one of the advantages is that there is more light for the leaves, because you're going towards the light. So there's more light for the leaves, and therefore more photosynthesis can occur. And the plant uses photosynthesis to get energy. Or food. And that gives the end to question 2. So again, the big advantages, of the fact that it grows this way is all to do with the fact that the plant wants to, even though the auxin grows away from the light, the plant itself wants to grow towards the light. So using this fact, the fact that the one side is elongated and it pushes the plant towards the light, helps the plant, because then it can gain more light as there's more surface area of the leaves are exposed to the sun. Question 3, student investigated growth responses in plants. The student grew a bean seed in a box filled with moist soil, as shown in diagram A. After the seed is started to grow, the box was turned onto the side and placed in a dark room, as shown in diagram B. Complete diagram C to show what the root and shoot will look like in 3 days' time. So what we have, plant grew a seed. So diagram A, we've got a shoot going up. So it's in a dark room. So what will happen is that the root will go down, and the shoot goes in the opposite direction. So we have a shoot that goes up this way. So they go in opposite directions. Roots will always grow down. Shoots will also always grow upwards. Draw a ring around the correct answer to complete sentence. The results in this case show that the root is sensitive to gravity. So gravity roots are gravity. Roots are in the soil. They grow downwards. They grow downwards because they are under the influence of gravity. If you didn't have gravity acting on it, then the roots will not necessarily go downwards. A hormone in the plant causes the growth responses. What is the name? So auxin is the plant one. Statins and steroids are not growth hormones in plants. Gardeners can use some plant hormones as weed killers. Give one different use of plant hormones by gardeners. So one use of plant hormones by gardeners is that they can be used for cuttings. In fact, there's quite a lot of reasons. So the first one really that we've got is that we can use them for cuttings. So we can use cuttings, or growing cuttings, we should say. And essentially, if you add rooting powder, which contains the auxins, they will produce roots and they start growing as new plants. So essentially you can clone plants using cutting. So you cut off a part of the plant, you put it in rooting powder, which has lots of auxins, which is a growth hormone, and then you get roots will form in the thing from the cutting, and therefore you can grow a new plant, which will be a clone of the plant that you've cut it from. You could also, another thing that you could look at, because another type of it is inducing flowering, and this is for Gibberellin, and that's another use that you could have for plants. So Gibberellin induces flowering in a plant. Again, this is because plants require a certain condition to flower. Gibberellin helps the plant reach those conditions, and therefore they can grow. Essentially, it's like a shortcut. Essentially, again with hormones, you can think of plant growth hormones. You can think of it as muscle building for plants. Essentially, if you take certain hormones, you can build muscle. So here you can think of it the same way. It's not quite the same, but it might help you think of it the same way. But flowering can occur. Another one that might happen is that ripening. So ethene is used for ripening, and that is another example that you could use. Okay, selective weed killers only kill some plants in the garden. Killing weeds in the garden reduces competition between plants. The three factors that they compete for. So one of the main ones that they compete for is light, because they're looking for photosynthesis. Every living thing, or the majority of living things, compete for water. And then plants, another one you could use is nutrients in the soil. Another one as well, very common, is space, particularly for plants. If you think about it, the rainforest, there's a consistent battle for space. So plants want to spread out as far as they can so that they can get all the nutrients, light and water. So there's always a battle for space, so that's what plants will compete for. But the key one really is light. You don't say the sun. Really important that you don't say the sun, because plants can grow without the sun in artificial lighting. So plants are actually competing for the light. Gardeners sometimes use weed killers to control the growth of plants. Gardener wanted to get rid of daisy plants growing in the lawn. They investigated the use of a weed killer. So the gardener recorded the number of daisy plants growing in different 10 metre squared areas of the lawn, made solutions of weed killer, which had different concentrations. So they put 5 decimetre cubed of each solution on 10 metre squared areas of the lawn, then recorded the number of daisy plants growing in each area after 2 weeks. And we can see that on the graph. If concentration of weed killer, we had 0, which had 8 had no effect. Weed killer 20, so 6, 8, 40, 9, 6, 60 units, 5, 2, and then we can see that with the 4 and 8, all essentially the daisy plants die. There's no daisy plants essentially after 2 weeks using a very concentrated weed killer. So give one variable the gardener controlled in this investigation. So we have to control some variables. So there's lots that you can choose. The type of weed killer is really important to keep the same. So the type of weed killer. Again, you only need one, but there are lots of them. The same volume of solution. So again, you're just changing the concentration, but you're not changing the volume. Because if you change the volume, then that will affect the results. And you haven't got an accurate way of comparing the different concentrations. Again, the same area. So we use the same area for it, and also the same time. So any of these will count. You only need one, but again, any of those are perfectly fine. Avoid talking about the concentration of the weed killer. You're looking at the solution itself, because the concentration of the weed killer is changing. Okay, so the concentration of the weed killer is changing, but the amount that you're using, remember the difference between the two of them. So you're still going to be using, for example, 5 decimetre cubed on each single one. It's just that some 5 decimetre cubed has more weed killer atoms inside, therefore has a higher concentration. And again, the number of daisy plants is not kept consistent. You can see 8, 6, 9, 5, 4, 8, so don't use that as one of your variables. Well, that would be incorrect. The gardener decided that the result for a concentration of 20 arbitrary units of weed killer was anomalous. Suggests why the gardener decided this result. So why? Well, because more grew after using the weed killer. And that's the anomalous. So it's the only one that had more after than before. Even the one that didn't use weed killer did not gain any more daisies. Why did the gardener put 0 arbitrary units? So why did it use the bonus? So it can be used to compare to other areas. It could also be as a control, but to be careful, not as a control variable, but it's as a control. So it's used as the control, but not the control variable, because the variable is changing all the time. The gardener concluded that the best concentration we could use all over the lawn is 100 arbitrary units. Suggest why the gardener cannot be sure about this conclusion. Well, if we look back at it, it's clearly not the best one necessarily, because we've got 80. We're not sure here because we've only got 4 daisy plants here. We don't know if essentially, if it had 8 here, does that mean that all 8 will go to 0, or only 4 will go, etc.? We can't really infer a 100% conclusion, so we really need to repeat the results. So the reason why is that 80 units, and you can use arbitrary units actually, also killed all. Okay, the next one then. Plants respond to different environmental factors. Describe how different environmental factors affect the direction of growth of roots, direction of growth of shoots, and your answer should refer to the role of plant hormones. Again, 6-mark question. Very, very key one that we need to do. Again, it's 6 marks, so make sure that you are writing in a clear way, and you're including as much as possible. So the key thing here is we'll look at the direction of roots. So the direction of roots is controlled by gravity and water. So roots will grow towards gravity and water, or in the direction of gravity and water. The reason, so how does it work? So auxin builds up at the bottom of the root, due to gravity, and also due to hydrotropism as well. It wants to go towards the water. Main one you need to know really is the gravitational one. Due to gravity, therefore grows downwards, where the auxin is found. Again, auxin is the plant growth hormone, so where it is where it's found. Now here we've done a lot about direction of roots, but we've also got shoots. So the direction of shoots is upwards towards the light. And this is because, again, a higher auxin is found. Essentially, auxin grows away from the light and causes it to be top heavy, and therefore that's how it grows. So oxygen grows away from the light, which is basically the opposite side. And then, so therefore, so from the light, so it's on the opposite side, so there the elongation forces the plant towards the light. And that's it for this one. Okay, question 5. When a seed starts to grow, the young root grows downwards towards gravity, the young shoot grows upwards away from the light. Name this type of plant response to gravity. So this one is called gravitropism or geotropism. Again, it's just a keyword that you should be familiar with, so just remember it on a flashcard. So gravitropism, or can be known as geotropism. Please don't put, like, you don't need to put positive or negative. Well, essentially it's going to be negative, but it's gravitropism. So the young roots grow downwards towards gravity, the young shoot grows upwards away. Give reason why it's useful for the young root to grow towards gravity. So essentially, if it grew away from gravity, it would struggle to get water. So it can take in water. Taking nutrients in the soil. The further it goes in the soil, the more nutrients it can get. Or ions, minerals, salts, etc. And it anchors the plant. So again, it provides a base. If it's all very close together, then a wind can blow it over. If you spread out your roots here, then when it's windy, the roots will anchor, has a better anchor base in the soil, so therefore will hold the plant steady. The root grows down towards gravity due to the unequal distribution of a substance in the roots. Draw a ring around the correct answer. So this substance is auxin. Again, auxin is the plant growth hormone. Okay, part B, the drawings show apparatus and materials. What we're going to do with this is that describe how the students could use some or all of the apparatus and materials shown in the drawings to investigate the growth response of maize seedlings to light shining from one side. Okay, so what we're going to do is we are going to okay, so I'll do the answer in here because then we've got the apparatus and we can see. So this is the answer to part B. Again, another 6-mark question. So here we're looking for how we're going to get the seedlings to do it on one side. So what I would first of all do is, let's get your box and use the scissors to cut a hole so that we can get, so to cut a hole in one side. Again, if that's the first step that I would do. Number 2, I would then use the scissors. So again, because it's asking you to refer to the materials, please be very basic. We're going to use the scissors to cut some tips from the shoots. So that means cut the tips from the shoots. Number 3, so the forceps is used to handle the seedlings, and we can place in the box. Number 4, we've got the ruler, and the ruler is used to measure before and after. So again, you want to measure this in an experiment. You're measuring exactly you want to measure the effect of it. So you need to measure the before and the after, and you're measuring the shoots, how big do the shoots get, etc. So measure before and after size of the shoots. Okay, we've got a lamp, so we're going to use the lamp as on the side of the hole. And this will create your one-way lighting because you've got one side. So again, our experiment here, so we've got our box, and we're going to put our hole here, and we're going to put our lamp, which is going to shine in here, and then inside the box in green is going to be your seedlings. So you can see that the seedlings only experience light on one side, they don't experience light on the other side, so you get this one-sided lighting effect. Again, so some of the key things that we're going to do, so now we've got our experiment set up with the lamps, etc. We'll put the seedlings in the petri dishes as well. They didn't ask for every single item, so don't worry, not all of it's used. So key things for an experiment: we need to control other factors that might affect our one. So for example, water, because plants, they dominate. Water plays a critical part. So we need to keep the water the same. We need to keep the temperature the same as well. Whenever we do any experiment, you should repeat. So there should be repeats of the experiment. So again, whenever you've got an experiment description, how can you repeat this experiment? And you should be able to repeat nearly all the experiments at GCSE. And again, that should be a key part as part of your method and experiment description. You should also control the dark, so we should have a control so we can compare. So this can be either complete darkness, so no light, or we could have all-round lighting. So I mean, that's another way that we could do. So we place it and we place lights all around it. So again, here we're picking up loads of marks. In terms of 6 marks, you're just trying to ram as much as you can. So what else are we looking for? Describe how a student used some of the understanding words, so you should include a description of the results you would expect. So again, the results, make sure you include everything. So currently we don't have the full marks, but the results that we would expect is that the one-sided light would bend towards the light. If we have, for example, the control, and the control here should be vertical. And that would be enough marks to collect 6 marks. So again, it's quite detailed in terms of, particularly when it's asking to use equipment, try and use as much equipment as you can. Explain how you're going to use that equipment. And then think generally about your experiment, like what are you going to control? Are you going to repeat the experiment? What factors are you aware of that might affect this experiment? So the more you put down, the more higher chance you are going to get more marks here. But that was a tricky question, and it's worth 6 marks. Part 6a, plant hormones are used in horticulture. Name one plant hormone. The obvious one that we should use is auxin, but you can use gibberellin, etc. as an alternative one, but auxin is the main one that we should use. Diagram shows how new plants are produced using tissue culture. Tissue culture is a type of asexual reproduction. So we can see here that we've got a potted plant. We're going to cut one of the leaves off, scrape a few cells off, and place it in some jelly with some nutrients and hormones. Eventually, what will happen is that there is enough of particularly there should be auxin there. If there's auxin there, then the plant will grow and will form a clone of that plant. So one of the key things is asexual reproduction. So the key main one of this one is that there's only one parent because it's a clone. There's no male or female, it's just one parent. and there is no mixing of genetic material. It is just one type. So no mixing of genetic material, so therefore there is no genetic variation. In fact, this is worth 2 marks, just saying this point, so therefore no genetic variation. And one other thing we could talk about is fertilization, or there's no fusion of gametes, or no meiosis. So that's another point that you might have. So there is no fertilization or anything to do with gametes, or a fusion of gametes. You could also talk about that they're genetically identical as well as an alternative for the third point. But yeah, 3 of these points and you get the marks. Another method of producing new plants by taking cuttings suggests one advantage of using tissue culture and not using cuttings. So tissue culture, so cutting will only produce one plant. What we can have for a tissue culture is that we can have many plants can be produced, or offspring. Again, we're taking the seeds. But avoid talking about how the actual plant itself, like the damage to the plant or anything. But it was allowed, it does less damage to the parent plant by taking a few cells rather than like lots of cuttings. Okay, the final question. Charles Darwin investigated tropism in plants, and some students did investigations similar to Darwin's. The students grew seeds until short shoots are grown, use a black plastic cover to cover parts of some of the shoots, put the shoots in light covering one direction, and put the box over. The diagram shows how the investigation was set up, and then we can see that the results as well. So part A, we can see that it grows to the right. We can see part B also grows to the right. Part C goes straight upwards. Part A just talks about, so again, why does this happen? Well, why is because auxin is produced at the top of the stem. Okay, so for part A, again, and for part B, auxin is produced on the side, that elongates that side and pushes them towards the light. For part C, the auxin is cut off, essentially. There's no, the auxin doesn't see the light, so if it's growing, it will just grow straight upwards. So in terms of good two barrels that this should control, so anything including, so first of all the size of the shoots is important. So you want equal length shoots, the light intensity, because you're using light, so you want to keep that constant. The same plant, or the same type of plant. And things like water, temperature, etc. So a couple of marks there. So yeah, just think about, what do you want to keep the same? Well, you want to keep the same plants to make sense, right? Or you want to keep the same amount of light, or something like that. Shoot A bent towards the light as it grew. So again, this is just carrying on from my explanation of before when we introduced the experiment. So what happens is auxin is a growth hormone. That grows at the end of shoots. So if it goes at the end of shoots, so auxin grows on the darker side. Or is found, maybe not grows is actually incorrect, so auxin is found. On the darker side, this creates an uneven distribution. Therefore, plant grows more on the dark side because that's where the uxine is found. And pushes it towards the light. Okay, what conclusions can be drawn from the results about the detection of the light stimulus? So the detection of a light stimulus is, so what conclusions? So it's on the tip or the end. Again, if we look at part C, we can see that it's covered, and therefore it's not growing towards the light at all. So the light stimulus essentially is on the tip of the shoot. Where in the shoot the response to the light takes place? So it takes just behind the tip, because it's not right on the tip, because that's where the light is shining on. But all the uxin is all here, all produced at the top of the tip, and it's all there, so that pushes it round towards the light. You could also say allow at the tip, etc. That's fine. And that's it for this set of questions. If you have any questions, then please do let me know.